Hi, I'm Hiroshi Yamakawa. I'll give you presentation entitled Towards a Qualitative Evaluation of Biological Plausibility for Brain-Inspired Software. As background, why biological plausibility should be assessed. First, brain-inspired software is significant in 3 points. High affinity for people, a promising way to build an artificial general intelligence, and vessels for mind uploading. Second, the advances in neuroscience and deep learning made to build brain-inspired software. Then, we would like to ask, is biological to lower the barriers to entry for such software development? Our non-profit organization promote to develop a brain-inspired artificial general intelligence by mimicking the architecture of the entire brain. We conduct this study primarily pursuit the second significance. The amount of papers and data in the field of neuroscience is vast. Through them, it is not easy to estimate the degree to which the software implementing the algorithms is consistent with the truth of neuroscience. How can we overcome this difficulty? Importantly, the range to which brain-inspired software should mimic is limited scope of neuroscientific knowledge. Intuitively, we want to take advantage of the findings, which will undoubtedly affect the computational capabilities of the software, but not in too much detail. It is specifically a mesoscopic level of brain architecture. This is, for example, the granularity at which similarities between convolutional neural networks and visual cortex or reinforcement learning models and basal ganglia circuits appear. So, the bridge between algorithms and neuroscience should be built on a mesoscopic brain architecture. We defined a brain reference architecture as a static reference model that describes mesoscopic level architecture of the brain. It consists of circuits and connections between circuits. As function hypotheses, each circuit has annotation of function and each connection has annotation of meanings of signals. We have defined the minimum description unit called a uniform circuit. Each of uniform circuit is neural population that transmit a computationally identical kind of signal. We have defined the brain information flow, BIF, as a format to describe a BRA. Please refer to the poster presentation by Naoyu Arakawa in this 2020 APCSN conference, for detail. Brain-inspired software can be described by component diagrams. And the descriptions of the BIF and that of component diagrams are almost same. Therefore, we can evaluate the biological plausibility by comparing the component diagrams reflecting the software with the BIF that summarizes the neuroscience findings. Because component diagrams and BIFs both represent the flow of information, we can compare them. In a word, circuits on the BIF can be mapped to components on the component diagram and connections on a BIF can be mapped to a port on a component diagram. So, we can assess fidelity value, that is the degree to which the software associated with the component diagram reflects the BIF. Then fidelity value could be defined based on a similarity of optimal aligned BIF and component diagram. What kind of processing can calculate fidelity value? Because the step 1 in the evaluation process is to determine the optimal alignment between the BIF and the component diagram by performing a comprehensive set of 3 comparisons. First, the semantic identities of functionality on circuit and function on component are compared. Second, the semantic identity of the function on connection is compared to the meanings of the signals on the ports. Thirdly, the mapping between the BIF and the component diagrams as a graph structure is evaluated. The remaining differences in the optimal alignment thus obtained represent a lack of fidelity. In step 2, the fidelity value can be calculated by using some function that transforms the amount of difference. We investigated method for assessing the biological plausibility of brain-inspired software. This method uses the brain information flow which describes the mesoscopic level architecture of the brain. In the future, evaluation methods should be refined to be quantitative and eventually automate. Various issues remain to be addressed as follows. Thank you for watching.